Hello everyone, welcome back to the final section of chapter 3 that we're going to cover. So anything that's covered in chapter 3 is fair game for the midterm on Friday, October 2nd. So these will be the last video or two that you will need to watch and study before your midterm. So for the last part of chapter 3, now that we know how neurons work, now that we know how the brain is organized, now we want to kind of understand, well, how do people study the brain? Obviously, you can't just observe the brain working as it is normally, unless you have X-ray vision, in which case, super cool. So how do we go about studying the brain? So there's a few different methods of study. The first one is postmortem studies. So these are pretty common for some case studies or for studying some overall structures or malformations in people. So obviously we're not just going to take a brain out of someone living's head and study it. We're going to wait till they die. There are some limitations, such as the brain starts to, degrade pretty much as soon as someone dies. So there's a, pretty short period of time in order to study the brain. And obviously we can't tell how the brain works just by looking at a dead brain. The second type of study is studying some live non-human animals. So this would be usually what we think of when we talk about animal studies. We're talking about doing some sort of methods that can include lesion study. So that's where you go in and remove part of the brain. We can do some genetic manipulation in animals. We can do some in vivo research, which is single cell recording work in animals. And so this way, then we can see kind of how does behavior differ when we remove a certain part of the brain, or how does a certain cell react during different behaviors. So that would be with those in vivo recordings. And then the last section or type of study is studying live humans. So we're going to go over a bunch of the different types of human techniques, but the first type is surgical techniques. So again, this is usually done in a case study setting, usually to kind of help a pre-existing condition, and then there's some side effects that we can study to look at human behavior. So that would be like split brain. That split brain that we talked about before, and there was that fun video. So they will cut the corpus callosum, usually to help in epilepsy. But then as a side effect, then we get the split brain effect. So we can study how the brain kind of works together and separately as two different hemispheres. And then the final kind of main method of study is brain imaging. So there's multiple different types of brain imaging, and we will go through them all kind of step by step. So here's a list of the brain imaging techniques that we use. We use them mainly in humans. There are some we can do in animals as well, but since we can do them in humans, it's usually easier, and we get more information from human studies than animal studies when talking about brain imaging techniques. So there are, like I said, a lot of techniques here. You can see that there's some in bold. So the ones in bold, I'd say, are the most common. So we're going to go over these 4 up top, and you're going to want to know the pros and cons of each. And then also this one down here, which is EEG, is a little different than all the others. And then we will talk about these non-bold ones as well. So you will need to know them even if they're not in bold, but the ones in bold are most common. So the first one we're going to talk about is EEG. So EEG is electroencephalograph. Really, we just call it EEG. And what this does is it measures the electrical activity via electrodes outside the body. So this is very non-invasive, and so it can be used a lot. It's also great because you can do it in both awake and asleep patients. So you can study certain states of consciousness, and you can record electrical or neural activity in different parts of the brain based on where you put the electrodes. So usually then there's about 50 electrodes at a time that are recording. So you can see how different parts of the brain react at the same time to different stimuli. So usually this is intracranial, so outside the brain, but there are times where we'll do intercranial, which means inside the brain, or inside the skull. But again, usually it's outside the skull because it's less invasive. And it has very good time resolution. In other words, it shows us the activity right at the time of stimulation. So there's no lag period. So that's a very good pro. Obviously, a con is you can't see the brain itself. So you're only measuring really electrical activity via the skull. So the first of the actual physical brain imaging, so where we're actually looking at the brain itself, is going to be computer tomography, or a CT. So you'll probably have heard of a CT scan in either if you've had to have one at the hospital, or if you watch medical dramas on TV, then they often send their patients for CT scans. And it's really great because you can see the brain itself. So it's a picture of the brain based on differential absorptions of X-rays. In other words, it's kind of an X-ray for your brain, and different parts of the brain will absorb different amounts of the X-rays, so we get a great structural feature of the brain. So we can really see different structures of the brain. So like here, these dark sections here are ventricles, or fluid-filled areas in the brain. This gray part would be the brain itself, and then the white on the outside, that's your skull. So different parts of the skull and brain absorb different amounts of this X-ray. And it's great because you can do it at different angles. So here we're looking down on the brain, so that's a horizontal section. But you can also take images coronally or sagittally as well, so depending on what type of image you need. Now, one problem with this CT scan is that we can't see activity in the brain. So we can see the structures, but we can't see what part of the brain is currently active. So if we want to see what part of the brain is active, then we might use a PET scan, or a positron emission tomography. So this builds a picture of the brain using short-lived radioactive material. In other words, usually then the patient will take, they'll have like a Gatorade type beverage that contains some radioactive material. This material will go all throughout the body, but including the brain, so it'll pass that blood-brain barrier. And it will, you'll get more of the radioactive material in parts of the brain that are more active. So the red parts are more active, or they have more of this radioactive material. So this shows us a functional map of the brain. So we can see what part of the brain is more active. Now, looking at this compared to your CT scan, it's a lot harder to see certain structures of the brain. So while the PET scan's great for looking at activity or function of the brain, it's not great at identifying what structure is actually active. We can kind of tell what area generally. So like this is the posterior of the brain, so we know the back of the brain's more active. So probably that occipital lobe, but we don't know for sure. So that's one downside of the PET scan. Now, we do have more advanced imaging techniques as well. So the CT scan and PET scan are relatively cheap compared to an MRI. So an MRI, it stands for magnetic resonance imaging, and a normal MRI looks at structure, okay? But you can kind of, if you look at this image here, then you see it's a lot more detailed than the CT scan. But otherwise, it kind of does the same thing. So it still looks at the brain structure. Now, it does take a lot longer to get this image in the MRI than it does in a CT scan, but that's because it is getting more details and more information. And it does this using a magnetic field because there are some magnetic particles in your blood, like blood contains iron, so it's really measuring, it's using where the blood goes to reveal this structure. But again, an regular MRI only shows us structure. It does not tell us activity or function of the brain. It's also very expensive, and it takes a lot more time than, say, a CT scan. So going from a normal MRI, then we have an FMRI, which is a functional magnetic resonance imaging. So this is where we add on that learning about the function of the brain, so what part of the brain is active. And it does this by measuring blood oxygenation and flow. So it measures the amount of oxygen in the blood and where that blood is flowing to. Because parts of the brain that are active require more oxygen than parts of the brain that aren't. So by measuring where there's more oxygen versus where there's not, we can identify parts of the brain that are active whereas are not. So it can produce pictures of blood flow in the brain taken less than a second apart. So it is a bit quicker. And so if you take an fMRI, which really an fMRI is only going to be this colored part, but what's great is you can do an fMRI at the same time as a normal MRI, and then you can put the pictures on top of each other. And that allows us to identify not only the area or what area it is, but also how active that area is. So by combining an fMRI with a normal MRI, we can get a full picture of brain activity. Now again, an fMRI is very expensive, more expensive than a normal MRI, and so that's why it's not done all the time. And also the machines for an MRI are quite large, and so not as many places have access to MRIs. So those are the four big, most common imaging techniques: a CT scan, a PET scan, an MRI, and an fMRI. Now there are other types of imaging techniques that you'll want to know. Personally, this one's my favorite, the diffusion tensor imaging, mostly because it creates really pretty pictures. And what it does is it builds a picture of water movement in the brain using an MRI scanner. So it's kind of like an fMRI, but instead of measuring oxygen, it's measuring water movement. And instead of being able to measure kind of brain structures, what it does is it gives you an image of neural tracks. So it shows how things are connected. So while the fMRI and MRI and those other imaging techniques kind of focus on the cell body or the soma and the activity there. The DTI focuses on what's happening in the axons. That's why you get to see all the fun tracks. Now, the next type that we're going to talk about is the near infrared spectrometry, or NIRS, the NIRS. And so this technique measures changes in blood oxygenation. So it is similar to the FMRI, except it doesn't use an FMRI machine. What it does is it shines near infrared light through the skull, and by doing this it can detect attenuation of the re-emerging light. In other words, it's an indirect measure of brain activity, so you don't really get to see what's happening real time like you do with a fMRI, but it does give an idea of what parts of the brain have or are getting more oxygen than others. So it's not used as much, but it is still a good technique, and it is cheaper than an fMRI, so sometimes they'll use it if they just want to look at it, like in post-stroke patients. They want to make sure parts of the brain are still getting oxygen. Then they might use a NIRS instead of an fMRI because it's cheaper and quicker. And you don't really need to know about real-time information with stroke patients when you're just checking function. Now, the last type I want to talk about is a transcranial magnetic stimulation. So this isn't really an imaging technique. It's more a way to kind of study the brain in a different way. And so what it does is it induces electrical activity via a magnetic field. So it's kind of like a big wand of magnetic fields. It's kind of a stick with a circle on the end, and the circle is a magnet. And so when you put this magnet over a part of the brain, what it does is it disrupts the activity in that part of the brain. In other words, it kind of turns it off. So what we can do is we can actually do a lesion study in humans by kind of turning off different parts of the brain for a little bit of time to study what happens to the behavior if that part of the brain is no longer active. Now this, it doesn't last very long. It does come back. Function does come back after, like an hour or two. So there's no real long-term effects. One study that has been done with this is letter detection. And so what they did was they had patients kind of try to detect or to recall letters. And when they put the TMS pulse to the visual cortex, what they saw was that people weren't able to detect the letters as much. In other words, they couldn't see the words or the letters because the TMS had kind of turned off that visual cortex. So they could still see, but they couldn't process what they were seeing. Because remember, the visual cortex processes the visual information. However, after a while, so after about 2 hours, then the activity came back up, so they were able to see again. So that's the last type of imaging technique we'll talk about, and that's everything that's gonna be covered on midterm 1. So if you have any questions, please send me an email, do the practice test, and really try making those glossary terms, as they'll help with your studying, as well as it'll allow me to see what type of information you guys are really understanding or maybe struggling on.